Okay, so thank you, Walter. It's an amazing meeting. Thanks, Marilyn. So I'm on page number 62. Looks like one can take notes. I thought that okay. So my original plan was to talk about one topic, but then when everyone got so excited about machine learning, I thought that let's start a little bit with that, and then move on to the topic that I originally thought about. Here is my team. So we are a fairly young team. It's like we're like 4 years, 4.5 years. I've just submitted my tenure package, and so some of the work that will be showing today will be by John, Jacob, Chun, and Eric. The first part of the talk will be Eric's work, the machine learning part, and then of course the ATP synthase part, which is more classical electrostatic, if I may, will be done by the three other colleagues in the group. Okay, so a little bit of a philosophy that we follow in the group is, most of the work that we see that has happened as molecular modelers, as our training, is the right-hand side of, actually, is it left-hand side of the screen. However, we really aspire to learn about diversity, which is a little different from complexity. And normally, this shake of hands is done by informaticians. You can always think of the Rosetta community, who are very, very interested in knowledge-driven methodologies. Can we actually think of, as physicists, is there a way of traversing this bridge? Of course, we'll use knowledge and data, but as physicists, are there certain properties that can be learned from a huge number of proteins and that can be used to study diversity? We think electrostatics can indeed be one such property. So that will be the first part of the talk. I talked a little bit about this last year virtually, but I thought that because machine learning is kind of important, so let's start with this this aspect of it. So with that, the first application area that, and again, we are a very science-driven team. We often use algorithms and methodologies by several people who are around in this room. And then we, start seeing, start making scientific discoveries. We have some methodology developments, but that's not in the area of electrostatics or free energy, so to speak. So, okay. So what we have come up with is a way of learning electrostatic potentials for protein peptide complexes. And that's the first part. I'll take you through that. So the example that we will learn about is, someone is moving the mouse, okay, is MHC protein complexes. So MHC protein complexes are, just to a little bit of a refresher on the biology of this. So let's consider this to be a cell surface. Let's assume that this is an infected cell. So any protein that you see in the PDB, all the structures, what happens is, after the protein does its function, it goes through the proteasome and gets chipped into very, very small peptides, peptides of the size 9 to 11 residues. So any protein, any structure, be it our own protein or someone like, or an external protein, that ultimately becomes very, very small peptides. So these peptides then gets, this is a small movie of the proteasome. These peptides gets carried to the surface of the cell. First of all, these peptides go on and they bind to a protein called MHC, or major histocompatibility protein. So these peptides then make this peptide major histocompatibility complex. And this peptide major histocompatibility complex thereafter goes to the surface of the cell, where they make what you call an immune synapse. So T cell receptors come in and make a complex with this peptide MHC protein. And now turns out, if these peptides belong to us, if these are self-peptides, if these are from the proteins that we have, then TCR will not bind to the peptide MHC. However, if these are external, if these are non-self proteins, if these are peptides that are coming from non-self proteins, then TCRs actually make this immune synapse. And after that, essentially that cell gets labeled by your immune system, and then this T cells, uh, they release a chemical called porphyrin, which actually now uh, goes ahead and they make holes in this infected cell, and your infected cell is destroyed. So that's kind of one of the pathways for gaining immunity. So your T cell is recognizing this cell and it's destroying it. So now think about it, then this recognition process depends very much on this peptide MHC complex. So you can now think, uh, so that's an important, and again, this is an example of a catch bond, but that's not important for this meeting. So now let's look into this peptide MHC complex. This is, of course, the background is the MHC protein, and you can safely think there are millions and millions of peptides that gets generated in our body every day. And these proteins that we have, the MHC proteins, are known to be the most polymorphic protein in the human proteome. So there are 20 greater than 20,000 variants of this protein, of this proteome. What is the world population today, roughly? Any guesses? 8 billion. The world population is 8 billion, and you have 20,000 variants of this protein. So of the order of, let's say every billion is 10 to the power 9. So 10 to the power 5 people around the world carries one version of this protein. In fact, each one of us has around 4 to 5 variants that we carry of this protein. So you can safely start thinking of, can we cluster the world population based on the properties of these proteins? Safely speaking, you can do 20,000 odd clusters of the world population. Okay, so now again, we are seeing, of course, these proteins have immense diversity. The region in the center is most conserved. As we go out, that is where the diversity is more. But the structure, remarkably, is very, very similar across most of the variants that has been known so far. Okay, so why is this so important? The reason this is so important is because I talked about the self versus the non-self idea. So if you know what are the immune peptides of this particular protein, then in principle, you can deliver these peptides to a patient. And this is something called T-cell therapy or immune therapy. And these peptides are, of course, they will lead to immunization. So there is a lot of work that goes towards predicting the peptide of a certain MHC protein. So given a patient, you can figure out what is the MHC composition of that patient. And based on that MHC composition, you can actually give them certain kinds of peptides, which will allow, think of it, a peptide means the T cell will come and break and essentially break this cell apart. You're actually inducing more immune response in that patient over what they have. So you can do that. So so there's a huge demand for creating this kind of therapeutics. So so far, most of the work that has been done is pretty much sequence or informatics based. So can we do something that we like doing, namely to start doing things with electrostatics? So the first thing we did was these proteins that you see, you can align them by the sequence, and you can find something called a Hamming distance. And you can see how does the number of mutations change with the basically the number of mutations. Oops, sorry. The number of what is that like? Don't worry about the construct of this graph. It's trying to show that if you are diverse in the Hamming distance, does that track with the diversity of the mutations? If you're, the next thing is blossom alignment. Blossom alignment is alignment of sequence together with conservation of certain positions. Essentially, you align sequences, but say that certain positions will have certain kind of residues. You see, like they track okay with the divergence of the protein. Finally, what you do is you do an electrostatic similarity index. We know that how to compute. You can create electrostatic potentials for the number of structures that are known between these. And you can see how different is the divergence between the sequences versus versus the deviations in the ESI. And you find out, okay, it's like 35% correlation. This somewhat of a divergence. So there's somewhat of a correlation that electrostatic properties of these proteins are actually changing with the sequences of this protein. Okay, so with that then, can we now formulate a tool, a machine learning tool that can actually learn the complementarity relationships between the protein and the peptide? How do we do that? Okay, first thing, when you start doing learning, we can't learn all the variables. We need a latent space. If I show you a protein in this form, you will not be able to even understand that it's a protein. Only in a reduced space of a ribbon diagram would you recognize that it's a protein. So when we want to learn something, we have to go to reduced spaces. So the kind of reduced spaces that we have created is using an architecture called the Inception Network. It's a convolutional neural network based on the movie Inception, the famous movie. It's essentially like a replica exchange molecular dynamics, but each replica is actually a convolutional layer. So it's a multi-layer convolutional neural network that we have chosen for doing a training. So what do we train? So we have the peptide MHC complexes. We have chosen 8,000, not the entire 20,000 variants. Of these 8,000 variants, there are 131 PDB structures. For each one of these PDB structures, we constructed the electrostatic potential using nonlinear PB. And then, nonlinear Poisson-Boltzmann equation. And then we chunked out, wait a second, yeah. Then we chunked out 3 1-angstrom cubed volume, literally the volume data of these. And we train that against the known binding motif of the peptide. So this binding motif is nothing but a alphanumerical signature. It's actually a probability distribution, if you can think about it. Logo plots are nothing but probability distributions. There are 9 peptides in one residue, as I mentioned already. Each position has a probability of the corresponding amino acids. And so, given the electrostatic signature, what we are trying to learn is using KL divergence as a loss function. If the protein has a particular electrostatic signature, what will be the corresponding peptide probability distribution? So we have 131 structures for which the binding motif is actually known. These binders are millimolar and or lower, millimolar or higher, essentially. So these are strong binders. So once we do this training, very good. So then the first thing we do is we may start making predictions. So as I told you that there are 8,000 variants we want to work with, of which we have used 131 in the training set. Now, of course, what happens to the 8,000 minus 131? Fortunately, we have AlphaFold or Rosetta Fold right now. So we can create the structures using AlphaFold or Rosetta Fold, create the corresponding electrostatic potentials, feed those potentials inside this neural network, and get predictions of the corresponding peptide binders, of the peptide binding motifs. So in this way, for all the 8,000 variants of this MHC complex, we can actually now predict what is the corresponding binding motif, what is the probability of binding of different amino acids. So that's essentially the sum of the neural network. So so we are training an electrostatic property with a probability distribution, and then using electrostatic properties of the remaining part, which is like more than 90%, we are being able to generate logo plots or probability distributions of unknown MHC complexes. So as a test, what we did was these probability there's a score, PWM score, doesn't matter for the time being. So what it tells us like how probable the binding motifs are. So it turns out that for going beyond the training set, for completely unknown data, with R value of 60%, 62%, we managed to track well, that the predicted binding motifs managed to track well with the known IC50 values or the question. So yeah, so the larger the value, that means more stable the peptide is.
What is the kind of blue, line that goes?
Oh, yeah. So that's bad prediction. We did not look into exactly what was causing at that particular IC50. What was incorrect in that prediction? We should look into that possibly. But the point here is, I wanted to make is neither IC50s nor KD values were a part of the training of this algorithm. But the most probable binding motifs that the algorithm produced were those that in fact had longer lifetimes or more stable IC50s. Then, of course, what next? Okay, now then.
Sorry, Hadi.
Question.
Sorry. No, coming back to what we were saying, you didn't train which was the target of your, how do you train your neural network?
So the inputs of the neural network was the electrostatic potential,
Yeah.
and the probability distribution on the right hand side, the probability distribution of the amino acids. That's it. So a spatial feature and a probability distribution.
Oh, but well, the probability distribution is somehow linked to the affinity.
And that's what, but we did not explicitly put the numbers of the affinity.
Okay.
And turns out that once we make predictions for the ones where the affinities were known, this tracks well with that. It correlates well with that.
Okay. Okay. Thank you.
Abhi, so it seems like the electrostatic potential on the surface would be the most important, and you might have is this also assuming that the peptide is binding in a restricted area that's similar for all of them?
Yes, which is very true for MHC1. If you go to MHC2, that's what we are working on. There's dynamics in this problem. Once that happens, we can talk more during the round table. We took advantage of the fact that this is a very, very rigid system, but at the same time, it's very diverse. It's possible around all. Question.
Okay, explain, give a little bit details about the inception block. What's there? Because that's like the features of the network. So
That's correct. So it's so normally, if you think of a traditional convolutional neural network, what you do is of course you have this data, you end up loading this data as a matrix, and then that matrix kind of gets stored into, like, by the idea of convolution. If you know a prior, then you can bring down the matrix into a.
So that's like the surface potential.
Exactly.
masking,
So we make
which is treated as the image.
So we actually put the voxels at a given voxel. What is the electrostatic potential value? That's a part of the matrix, and the corresponding probability distribution. So the voxel that comes out of APBS, like a 1 angstrom cubed, voxel, the corresponding to that, what is the potential value XYZ? You put that in this.
Okay.
So you create a matrix, and that matrix kind of gets convoluted into a reduced dimension, and you learn based by choosing a loss function that for the data, again, for the 131 structures that is known, learn in a way that the distributions are closest to the experimental distribution, not using RMSE, but using Kane divergence, because distribution probability overlaps are best handled with KL divergence and not with RMSEs.
So what's the dimension for that inception?
Great question. So the dimension for that inception network is going to be 131 structures times 3, which is the electrostatics block per structure, times 9, which is the probability of each one of these each one of these binding motifs. Now, I should also mention that we have chosen the binding motifs in such a way that each one of this probability is constructed of 50,000 or more peptides. So what you see on the right-hand side is actually a representation of 50,000 or more. That's when you get a proper probability distribution of the peptides.
Thank you.
And one more thing, any problem due to the fact that the KL is not symmetric? or did you see?
Great question. So when KL divergence is not symmetric, it's correct. KL divergence is not symmetric. So what you do is a forward and a backward divide by 2, which is actually called a JS divergence actually.
So you corrected for this.
Yes. Yeah, absolutely. So, okay.
Sorry, one
Uh,
more.
Question. Yeah.
Sorry. Yeah, neat idea. This is kind of a inverse folding problem where you don't have part of the structure. But often in inverse folding problems, the sequence recovery is not that great. How good is, did you look at that sequence recovery at this model?
So we have to define the inverse folding problem a little bit better to me. So sequence recovery. So that's what I was trying to get to. The problem of doing something like this is that the ground truth, there is no ground truth. We can get new insights, and the value of the insights are what holds the benefit of this project, so to speak. So you're asking the question, once I make a prediction of this binding motif, what is its verification? The way we are thinking about it is, let me go through a little bit and then I'll tell you. Okay, so this was within in the realm of verification. We have also done cross-validation analysis. Cross-verification analysis, we took out 10% of the data, very standard things that I'm not showing today. But a very interesting thing came out, which is what I want to show you. So these peptide, these probabilities, if I may, this probability, let the binding motifs are probabilities. Now, as I told you, I can now construct 8,000 of these probabilities. And using KL divergences, I can cluster these probabilities. And all of the binding motifs of the 8,000 of the 8,000 MHC proteins can be clustered with some manual tuning to 12 clusters. What this means is the entire world population, all of their MHC complexes, when they are looked from the viewpoint of electrostatic potentials, are only of 12 kinds. Okay, so there are, of course, red means each line here means KL divergence between two different kind of clusters. Larger values are red, smaller are yellow. So what does this mean? We were trying to understand what does this mean. Again, I have no way of validating this. What does this mean? So then we have colleagues at Mount Sinai who work on something called immune checkpoint inhibitors. So patients of cancer are given certain kind of medication to see if their lifetime improves. So what we found was that patients who are actually living longer, who are kind of responding to this medication, are those patients that has 4 to 6 of peptide binding motifs that are most far away from each other. Patients with the most diverse repertoire of MHC complexes in the space of electrostatics are those patients that are surviving longer. It's very, very empirical. There is no justification to this, but at least we found something like that. Another thing that we found is very interesting. Okay, this is a very, very fast code right now. The entire human proteome can be done in 7.73 seconds. SARS-CoV-2 can be done in less than a second, once the training is done, of course. So if you see the number of predicted peptides that we have, self versus non-self, now what we can check is, of the entire human proteome, the electrostatic properties of the peptides that we have versus an external peptide. HIV peptides in the electrostatic, with an electrostatic lens, look very similar, like more similar to human peptides over something like influenza or SARS-CoV-2, in fact. So this, again, looking into using machine learning, using electrostatics as a loss function in machine learning, one can look into peptide properties and see that their properties are similar or dissimilar between humans and non-humans. So again, coming back to the question of validation, it's very difficult. What we do is, fortunately, we have experimental colleagues who trust us. So then we say, okay, if they have, we work at, so I also work at the Mayo Clinic. So often they have cancer mutations. and they want to know what is a peptide target for the cancer mutation. So these kind of tools are used to make those predictions. And then ultimately this is going to work in the experiment. Who knows? 60% of the times will be work. But at least it's better than zero, infinitely better than zero. So that's kind of, again, a way of utilizing all the intelligence of this community. Let's say the better electrostatic potentials we generate here, it will go into this this machine learning process. The better we know how to compute pKs, for example, but these are surface values, so might be we were not terribly bothered about it. But I wanted to kind of tell you that how we can use electrostatic potentials. So this paper is now okay. We have a website. So this paper is now under review with Cell Systems. We actually have a website where you can go and you can actually put in the sequence of any protein that you want, and this peptide will spit out for you what are the possible immune peptides from that protein. So a test case is, if you put sequences of wild type proteins, human proteins, we will not be able to predict a single immune peptide because our proteins do not work against ourselves. But if there are cancer mutations in the proteins, those kind of peptides will be predicted from this website. And then again, so that's some sort of validation. If you have for example, we predicted any kind of epitopes. So SARS-CoV-2, for example, had very known epitopes on the spike. So if you put the spike protein inside and you get it will tell you what is the peptide that comes out from SARS-CoV-2 spike. And those peptides match really well with 90% accuracy, so to speak. So for certain, so as a ground truth, for proteins from which immune peptides are known, like cancer mutations are often known, if you put them into this methodology, then the peptide outcomes match very strongly with known immune peptides. So that's one way. But again, a very, I would say, very offbeat way of looking at this area. But I thought that it's interesting. That we should not just stay stuck. So again, coming back to diversity, now think about it. Taking molecular properties, we're now looking into patient populations, and electrostatic is allowing us to do that. So I think that's something that I felt was very, very, very revealing about this piece of work. And we continue doing this. Our next work is MHC2, and yeah, okay. How much time do I have now? Let me check quickly because. Okay, 11:32. I have only 10 minutes for the real talk that I wanted to give. So, but okay, I will not give the real talk. So, okay, protein electrostatics in molecular bioenergetics. Let me skip all of these. So we, of course, are a molecular dynamic simulation team. We run a lot of, so we are developers of a part of a tool you might hear, NAMD. So we are partial contributors to development of NAMD in their in what you call the integrative modeling suite, MDFF, for example, if you've ever used, those are what I'm responsible for developing. So we had the audacity of simulating an entire cell organelle back in 2020, and now then a very key problem has come in the all of these. Of course, we do a lot of free energy calculations also. Fine. A very key piece of Rhodobacter spheroides someone was showing this morning. So this is a chromatophore from Rhodobacter spheroides. So ATP synthase is very close to our heart. The molecular dynamics of this motor is very something that we are very interested in. So very recently, there is a huge controversy in this area, so much so that NIH actually held a meeting to address this controversy, and in fact, come up with specialized funding calls for addressing ATP synthase. So what happens is, there is something called mitochondrial permeability transition pore. What happens is, as our cells age, the calcium flux across the cell, the cell starts losing calcium flux. And as that happens, you essentially end up losing the potential, the transmembrane potential, and the cell will have to undergo a stage of apoptosis. What is this transition pore is a matter of tremendous contention. There are 11 candidates, no less, of where the transition pore can happen, starting from VDAX, which is a voltage sensitive channel, to ATP synthases, to I've forgotten there's the list of 11 proteins. And some people also feel that it's a combination of a few of those 11 proteins. And but what is the molecular mechanism for that is not known. So I'm very interested in ATP synthase, and some empirical models of that is as let's say, I show you a cartoon of F-type ATP synthase. As the calcium flux increases, calcium comes in and binds to this part of ATP synthase. A disassembly process begins. Okay, there's another thing called a cyclophilin D that comes in. A disassembly process begins, and this C ring of ATP synthase, the FO, the lower part, very popular part, that opens up, and there is a secondary channel that forms through it, a porine that forms through it, which causes the cell to lose all its osmotic potential, and the cell dies. This is of course different from the primary channel that is somewhere here, which might be we have seen. So this is the presence of a secondary channel. And of course, where is the controversy? Not only that, so my colleague Nelly, she's used to be in Yale, now she has moved to Penn State. She has actually done conductivity measurements of just the C ring of ATP synthase, the secondary channel of ATP synthase. So there is evidence for that. But what is the controversy then? So one of the mechanisms is that magically the C ring expands. Like the disassembly happens, the C ring expands, water and iron goes through it, and you know, your pore gets made. Mechanism 2 through N, in fact, mechanisms every 2 or 3 months, a new mechanism keeps coming up, where essentially someone says that, no, this is not the pore, but ATP synthesis actually form dimers. And it's between these two, that's where a pore gets formed. In fact, ATP synthase now actually end up making super complexes. It's called a respirazome, and people feel that between them, pores are getting made. There is a mechanism X, which says that if F-type ATP synthase does not even work as a transition pore. So all of these controversies are present, and how can we work through these controversies is where we started. So working with Leo Sazonov, the first thing that we did was, Leo said that, Avi, you have to disassemble an ATP synthase. It's very difficult to disassemble an ATP synthase because going back, ATP synthase cyclophilin D complex is not known. It's structurally, it hasn't been resolved yet. So then how do you disassemble this? So what we did was we removed this OSCP completely from our construct. As soon as we did that, this peripheral stalk started doing pretty weird things, and a pore opened right through it. And if you see here, there is a secondary pore that forms purely through the disassembly of ATP synthase. So a simple mechanical push can open up this otherwise, you know, closed channel. In fact, there's an assumption in this model. Normally, the C ring has lipids inside. I have purposefully taken them out. But turns out that in the absence of the lipid, water can actually flow, go through this. Okay. This is, of course, a close-up of that. So this is the primary channel, which is at the interface of the C ring and the A subunit. And this is the second, which was very well known, like Wolfgang Jungi has done a lot of work in Germany on that. John Rubinstein nowadays is really, there's a lot of cryo-EM structure. But the secondary channel structurally was shown for the very first time that water actually goes through this. So the next thing is, okay, now we have taken a simple C ring, just a simple C ring, and simulated that across a range of voltages. And we found that the number of water molecules going through is the minimum at 0. It goes up more at minus 180. And of course, at plus 180, it goes up, but not as much. The radius does not change too much of this, but it goes through. Now, there is something tremendously wrong in the image that I've put there. So the reason for that is what I've done is I've taken all the waters and I've overlaid them. If you overlay waters in a channel simulation and show them, then everything looks conducting, which is actually not. So you should never do something like this. I just wanted to kind of, however, in contrast, you should do something like this, where an instantaneous snapshot of water goes through. So, okay, what we also found is that the number of water in this channel is mediated by protonation, deprotonation equilibria of certain acidic residues that are in the middle of the channel. So you have 8 glutamic acid residues, famous glutamic acids that are there. So how much water is present in the channel depends also on the protonation states of these. So it's a coupled problem of voltage, protonation states, and water coming in. With a system size of 500,000, I will not dare to do constant pH. So, okay, these are, of course, so we work with Marilyn on MCC. I cannot do that. Constant pH is possible right now on NAMD, but again, I will not dare to do any such thing. So I went the, the hammer way, essentially like the most brute force way of doing this is I took the channel and I deprotonated the channel from 0 to 8 and simulated each one of these channels from minus 180 to plus 80, plus 180 millivolts. Crosses means that the channel did not open. Ticks mean that the channel opened. And so I found her. So again, you cannot do these kind of things without a supercomputer. So it turns out that we have some of the big exascale computers in the country that allows us to do this safely, 100 microsecond worth of simulation on this slide. So what you see is that as the voltage decreases, the number of times, tick means that the channel has opened. The number of time the channel has opened, you only require 3 deprotonations to open this channel. However, as you go to lesser voltages, the number of deprotonations increase, and then ultimately it comes down again. So you actually can see what are the conditions under which this channel is actually activated. And now the next question is, does ions go through this channel? Can people guess what are the conditions under which the ions might go through this channel? There are only 2 points. There are only 2 points on this phase diagram where conductivity can happen in molecular dynamic simulations. So I'm giving you a channel that has 8 negatively charged residues in the center. And I am deprotonating all the 8 in this case. The channel does not conduct because sodium comes and just gets stuck on the ion. It's very, very difficult. So there are only 2 places in 1.5 microsecond I could do some conductivity measurement on this using traditional ways, and we got some sort of this. Of course, this is what you call what you call stationary current. This is not the kind of currents that we saw yesterday or Anna was showing. This is kind of a linear fit to that graph. So you get some currents here, and then this is kind of a linear fit to that. And then, okay, then you have gotten some currents, but what is the mechanism? I think in the last 5 minutes, let me tell you that, and then, yeah. So it turns out that human, bovine, or ovine have a helix that looks like a dipole. As opposed to yeast, which was done back in the day, there is no dipole in there. Or a plant, the helices are not a dipole. So what happens is, if you take a dipole and start simulating that in the presence of an electric field, it starts reorienting. If it's positively charged, if it's a positive field, then the reorientation is less, because essentially this is negative, this is positive. The field direction is such that it more or less likes to stay in that. However, if you flip the field, then your dipole would actually, your north pole would like to go to the south pole, and the south pole would like to go to the north pole. So you should see great rotations. So this now happens in the C ring. Now the C ring in the presence of, so our model is this dipole moment in the presence of an electric field reorients more in the negative field, less in the positive field. It's very unique to mammals, because mammals has that dipole in the helix. And once that happens, this thing opens up, and essentially this is the inner part of the pore. You start getting water inside. Not only that, a single helix, not the entire C ring, even a single helix can start leaking water under certain conditions. So with that, then I said, Nelly, Nelly, who's our collaborator, let's just disturb this dipole, break the dipole from the top, take the positively charged region and neutralize it. And of course, the conductivity came down. So did our number of waters. So this is kind of one way of looking at that. This is a small movie of how the water goes in, because there is already lipid. All the results that I showed you was without the lipid. Now, if in the presence of the lipid, the water comes in and displaces the lipid, the upper part is completely gone, but the lower part still remains. So then we did another free energy calculation to figure out what is the energy required to remove the lipid, because a pore cannot be formed before lipids come out. So the mechanism of the lipid exit was very, very important. We did ABF calculations for doing that and found out that single tail lipids are easier to remove than double tail lipids on this which was actually an experimental thought that Leo had. We verified that. So now actually we know that this pore can remove the lipid, water can go in, ions can go in, current can, like with some level of approximation, currents can go through because of the dipole moment. And then finally then, so we started doing a whole ATP synthase simulation. But okay, let me not get into that. Otherwise, it's going to be too much. But so the point is, in this preliminary work that took us 2 years, we have been able to open up a channel that was not opened so far, show that electricity current can conduct, and not only that, lipids can be removed under normal room temperature conditions with very low barriers to open that channel up. And how is it important in the context of the full ATP synthase mechanism? It'll take us a little longer. But okay, I think I'm over time. So yeah, thanks for your time.
Thank you.
Hi, thank you. So I'm really curious about that lipid, actually, how does it go into, it looks like it goes into water, right? Or, I mean,
This one. Oh, sorry, the one before. Yeah.
yeah, how do you remove the lipid?
It's just normal MD. That was so surprising that the upper part of this, that the upper leaflet gets dissipated right away. It's the lower leaflet where the lipid gets stuck. So if you see, like the lipid is, the water has come in. This by itself, the seeding is so hydrophilic. It loves water so much that water comes into the middle, no problem, even when the lipid is present. When the lipid is gone, there's a lot of water. But even when the lipid is present, it goes up to here. Now the problem is here. How is this lipid gone? Now, what we found is that to remove a lipid from here, if it's 2 tails, the energy required is more, which is the ABF calculation that I'll show you later. But if it's a single tail lipid, it's easier. Now there's a mechanism that I didn't get time to tell you. So on how to remove this lipid in the real ATP synthase, I'll show you afterwards. But yeah, like it is very interesting to see that the upper part, even if the lipid is present, and we worked really hard to get the density of the lipid correct here. So we did some, John Walker actually had a paper on the lipid density inside. So we were very careful about it. So it's not a artifact of putting lesser number of lipids, but yeah, so water just comes in very naturally in this pore. Yeah, so.
So I have a question on both sides of this. So I really thought the C subunit was quite hydrophobic, except for that one acid in the middle. And that when the acid is ionized, it rotates it 2 helices and it moves out into the A subunit. So why are there's so much water liking to be near?
Yes. So actually, this is what you mentioned is a reflection of people making interpretations of human systems based on yeast, which is something that we should not, as more and more we work.
So the sequences are very different.
Very different. In fact, that's what I'm showing you. So this is yeast, for example. Yeast is in the middle. Yeast is more hydrophobic than humans. So when simulations were done back in the day, they were done with yeast, and people were they were very, very confident on telling this is human outcome. But it's not. And like as biophysicists, we should be very, very careful about when we're working with human, we have to work with. And now with AlphaFold, it's even clearer. So that was the reason. The water, in fact, this paper, I am not telling the name of the author on purpose, but this paper is factually completely correct in that if you take this sequence and this seeding, you will not get a pore. But the problem is that's only for yeast. For humans or yeast or plant, for example, because both are more hydrophobic. Human structure just came in 2020. Before that, there was no human structure available.
I mean, it's also human, higher animals all
Exactly,
have
yeah.
these very small rings of only 8, whereas everybody else is 10, 12, 14.
That's correct. That's correct.
So on the MHC thing in the electrostatics, one of the things that Diane Barber taught me is that a lot of the cancer mutations,
Mhm.
huge number of them
Mhm.
are charge changes in charge.
Mhm.
So they're arginine to histidine or.
Correct,
glue.
correct.
So I'm wondering if that's perhaps one of the reasons that these are working. So
In fact,
well
that was the
with
reason we started out with that. But then I didn't tell you van der Waals are also important for proline. There are some peptides that has prolines in them. So we had to retrain this network with also a van der Waals map. And then it's all now it's there, like you can see it on the online. So, but electrostatic was something that was very, very, remarkably, we were happy. That very simple thing, just do APBS, do it on a number of things and you get these kind of signals. Yeah. So now we're actually trying to train electrostatics with phi and psi, some sort of orientations. Think about it, then you can predict Brownian dynamics with a tool like this. Then the outcome is going to be orientation as well as signatures. So yeah. So
If you included hydrophobicity complementarity in addition to electrostatic complementarity, would you see an improvement in the correlation?
I think so. I just don't know how to incorporate. Hydrophobicity is a very touchy thing, right? Hydrophobicity as van der Waals is the enthalpic part of it is okay, but I will never get the entropic part unless I get the entropic part, or you teach me how to do that. I have to compute so many free energies. But I must say, in principle, yes. So in Los Alamos, they had started a project where they said that we will get the free energies of all of these peptide MHC complexes. That proposed, I don't think they got computer time for it. But in principle, yes, from there one can get hydro. But that's why, like when you say hydrophobicity, the van der Waals part is simple. It's just shape complementarity. But the hydrophobic aspects of it has to be thought across a number of these things. Yeah. So might be now it's easier because I have 11 clusters. I can put one structure, a cluster, and I can do a little bit more. But hydrophobicity, normally I stay away from hydrophobicity unless Kendall or someone is helping me with that. So, so yeah.